So, hello everyone. Good morning, good afternoon, depending where you're logging in from. My name is Joanna Kutruba, and I'm a board member of the Young Researcher Committee, part of the European and International Zebrafish Societies. And together with the support of the ZDMS, we've actually come up with this FPK webinar series. Um, if it's your first time, just to have an idea, we're always showcasing two early career researchers that are sharing their scientific stories. And we have a Q&A session that takes place. And of course, the idea is to essentially get inspiration and maybe collaboration, feedback, and so on. Today's speakers are going to be moderated by the speakers of our first session that was held back in April. And for the ones that missed it, they can always find it and watch on demand on our YouTube channel. If you have been informed about this webinar through the IZFS or IZS email list, we are always present on LinkedIn and Blue Sky. So we would love for you to come and network with us and follow us either or. And with this, I just want to pass it over to the moderators, starting with Joaquín. So feel free.
Thank you, Joanna. It is my great pleasure to be here. My name is Joaquín. I'm connecting from Switzerland, where I'm a postdoc in the Biozentrum. Today, we're all delighted, I am, and we're all delighted to feature two great people, early career researchers. Each speaker will have 20 minutes, followed by questions and discussion. Um, Reminder that if you have a question, please type it in the Q&A box, and then we'll go through them at the end of the talk. It is my great pleasure to introduce the first speaker, Wei Jing, who is a researcher fellow in Gaurav Varshney's lab in Oklahoma Medical Research Foundation in Oklahoma City in the US. Wei completed his PhD in Peking University in China, where he used zebrafish models and genome editing approaches to study nucleolar gene function and endoderm development. Then he moved to the Varshney lab, where he focuses on developing CRISPR-based editing tools for precise in vivo modeling and functional interpretation of human genetic variants. Today, he will be talking about his work titled the Umax platform, a high-efficiency base editing toolkit for rapid functional assessment of genetic variants in zebrafish. And in a personal title, I've used the technology and I can tell you that it works like a charm, and I'm very excited to hear more about it. So please join me in welcoming Wei Jing.
Yeah, thank you for the introduction. Hello, everyone. Yeah, my name is Wei Jin from Dr. Varshney Lab. And first, thanks the community to give me the chance to present our recent work about base editors in Zebrafish. We built a platform called Umax, and this platform can be used to rapidly functional assessment of the genetic variant. So let's begin with a scientific problem. And we know with the complication of human genome projects, numbers of GWAS and genome sequencing projects come out and generated thousands of candidate disease genes and variants in humans. And among these variants, about 19% are single nuclear variants. So how can we determine that a mutation is causing a phenotype is a big problem. And as we know, most human disease candidate genes can't be proven with clinical or genomic data, so there needs model, especially in vivo animal model, and zebrafish is a good choice. And usually the first approach we think about is HDR. But HDR can be affected by many factors, especially in zebrafish, the efficiency is quite low, and has highly locus dependent problem. So we can't use this project to do variant assessment at a large scale. So our big question is, how do we functional validate a large number of single nuclear variants? So this where the base editor comes in. So what's the base editor? Actually, it's very easy and simple. And base editors can convert single base to another base in a target manner without double strand break. And now there are two main base systems. The first is called cytosine base editors, CBE. It can convert base C to base T. And second is adenine base editors, is called ABE. It can convert base A to base G. And in theory, these two base editors can address about 60% about the human snaps. And even the development of the base editor is about 10 years. They still face several challenges. For example, the reduced in vivo activity and the targeting limitation and bystander mutations. And this is the timeline of the base editing development in Zebrafish. So compared with hundreds of papers in human cells, actually in Zebrafish there are few papers. And next I will show you how our lab optimized this system in Zebrafish. And because AB system and CB system face similar challenges, so I will briefly cover the ABE and mainly focus on CBE system optimization. So for the AB system, actually the strategy is very simple. We collect the mutations from several well-performing ABE systems in human cells and to generate 10 new tools by combining the mutations in different ways. And in order to quickly and visibly to access the efficiency, we inject these things into the homozygous albino mutant. So we can easily to access the efficiency by the level of pigment rescue. And among the tools, we found the tool 3, which has these two key mutations, showed the best performance. And here we called this tool ABE Ultramax, short for ABE Umax. So based on this, we constructed several other tools which have different features, such as the shift window and precise window. And for the details, you can see our published paper. And next, I will mainly talk about the application of these tools. Since our lab is interested in the hearing gene, so we will choose some hearing genes for future study. And Zebrafish lateral line system is a very useful model to study the hearing loss because it shares the similarity with mammalian inner hair cell. So it's very easily to access the hair cell functions by using some live dye such as UPRO1. And the red bottom figure is a positive control. And you can see in the CDH23 mutant, the signal is absent. That means the hair cell function is totally affected. So I choose two missense mutations from these two genes, MYL7A and H1C, from the ClinVol database. And in the ClinVol database, these two missense mutation is labeled as likely pathogenic, but not validated yet. So after our experiment, we can see both mutations can result in the hair cell function phenotype. So for the genotyping, we can see the targeting efficiency was very high, and for this case, it's up to 95%. So except the coding region variants, we can also do some non-coding variant, such as splicing variant. So we designed a guide to target the tyrosine genes. And from this case, you can see the inject fish result in albino-like phenotype in zebrafish. And this case is exciting because the efficiency is about 100%. So after talk about the ABE, I want to mainly focus on our CB system. So both systems have the similar challenges, such as the hiding efficiency, the limited target window, and the precision problem. But compared with the AB system, CB system a little complex and has itself challenges. For example, the sequence contains bias and the high induce. So previously, the people will change the dynamics in the CB system and to update the system. And this situation changed because each dynamics has its own limitations. So we can't use it widely. So until 2023, three groups independently reported that TED-A derived CBE system can perform very well in human cells. So that means the strategy in the AB system maybe can be applied to the CB system. So after testing the three systems in Zebrafish, I combine the advantages of these two systems and introduce these two key mutations I identified from the AB system and to establish a new system we called ZTED-CB. So after testing multiple sites, we can see this system indeed showed no sequence content bias and reduced induce. But from the figure, you can see the average efficiency about 35%. So the efficiency is not high enough to do the F0 application. So we aim to develop a more high efficiency CB system. So I will talk about how we optimize this system one by one. So the first is the editing efficiency. We want use the system to do the bioalic-based editing in F0 Zebrafish. So inspired by the David Liu's group, they established the CBE6 system, which showed an enhanced CBE activity in human cells. So we optimized this system and introduced our self-mutations and to construct several new tools we called TCB 1.1 to TCB 1.4. So after testing multiple sites, we can see here this tool, TCB 1.2, perform best, the highest efficiency and the low induce. So here we named it TCB Umax. So after testing multiple sites, we can see the average efficiency is about twice than the previous tool, ZTED-CBE. And now the average efficiency is about 60%. And still this system showed no sequence content bias. So after first questions, let's move on to the second, the targeting limitation. And we know the CB system has two parts, the dynamics and the Cas9. So for the system itself, it has the target window. For example, here is from base 4 to base 8. And the second is the PIM requirement. So for the PIM requirement question, actually is very easy to overcome because now we have several new variants which can recognize different PIM, such as PLY, SPG, and NG. And from the figure, you can see all variants perform very well in Zebrafish. So for the target window itself, we use the strategy we applied in AB system. So we delete one part of the HNH domain and insert the dynamics into the middle of the Cas9, and to get this tool we call TCB Umax Extend 2. So after testing multiple sites, we can see this tool indeed shows the shift target window more close to the PAM sequence. Now the main target window is 12 to 15. So let's move on to the third questions, the induce. So not like AB system is very clean, and TCB Umax system actually can induce variable induce frequency. So here are three types, the low induce, moderate induce, and high induce. And as we know, the high indels will definitely affect our variant assessment, especially we use in F0 zebrafish. So how to reduce the indels? And a previous study have shown that the amino acid at this position is located on the surface of the Cas9 and is more close to the non-target DNA strand. So I hypothesized that if we insert the DNA here, maybe can let the structure more compact and to reduce the indels. And the result is positive. From the single result, we can see at some site with high indel efficiency, we can see this tool indeed can decrease the indels. So further quantitative analysis confirmed this tool significantly can decrease the indels. So finally the last question is precision. That means if the target window has multiple C, can we target the single C previously? So we still use the strategy from the AB system. Now there are several strategies can be used to improve the precision of AB system, such as introduce the key point mutations and delete the linker between the Cas9 and the enemies. So we use the same strategy to construct the 3 new tools. Here we call TCB Umax REST1, REST2, and REST3. So after testing multiple sites, we can see the result. Here, TCB Umax REST1 shows the shortest target window from base 5 to base 7. The original is from base 4 to base 8. And TCB Umax REST2 shows the best trade-off between precision and efficiency. So now that we have lots of tools, so how we use it? And so for first example, I want to show we can use this tool to do the functional analysis of candidate disease genes in the F0. So due to the time limitation, here I only show 2 examples. The first gene is called ETFA, which encodes a mitochondrial protein, which is essential for the lipid metabolism. We can see all the injected fish shows the lipid accumulation in the liver regions, which is consistent with the loss of function phenotype. And the editing efficiency at this site is about 98%. And the second case is MYHZ 1.1, which encodes a part of myosin 2. So the zebrafish shows the abnormal muscle development, and the efficiency is very high, and which means this missense mutation is pathogenic. So finally, we want to answer our big problem. So we want to know whether our system can be quickly used to access the variant functional analysis at a large scale. So we go back to the herring genes. And this is our pipeline. So after inject the related guide, we at 2 dpf, we will do the pulse genotyping and to determine the average editing efficiency. If the average efficiency is about 50%, we will move on and at 5 dpf, do some UPRO staining and behavior test to observe the phenotype. And then at 6 dpf, to do the individual genotyping to check the phenotype we observed was correlated with the edited result. So we choose 15 VUS sites from these 2 genes to test. And here is our result summary. And here we can see we can give you a clear conclusion at 14 out of 15 sites. And most of these sites, even we use alpha missense prediction, the result is ambiguous. So that means our experiment can give you a clear conclusion. So you maybe notice that these 5 variants are labeled by red box. That's because these 5 variants are induced by a new tool called TCB Umax 2. So what's TCB Umax 2? And the CB system contains 2 parts. The first is the DMAs, the second is Cas9. So we introduced some key mutations into the TET-A DNAs. So we hypothesize if we introduce some mutations into the Cas9, so maybe it can further improve the tool's activity. So we introduced these 2 key mutations, and the result is positive. At these 5 sites, the TCB Umax 2 can increase the efficiency at a different level. And here are the UPRO staining results responding to the 15 VOS target site. And we can see different misses mutations have different lows of impact. So now we have lots of tools. How can we choose the best one? Actually, it's very easy, depend on your sequence and your purpose. So the first thing you need to check is the sequence around the target base. If the sequence has a NGG, you can choose the uppercase. If not, you will use this variant, SPRY, SPG, and AAG, because it can recognize different PAM. And if your goal is efficiency, you can choose the up branch. Try these different tools. If you found your site has lots of indels, you can use TCB Umax Extend 1 to avoid indel generation. Otherwise, you can use these 2 tools. So if precision is your goal, you can use these 2 variants. So finally, I will give a short summary. First, we built a platform, Umax. It can improve the efficiency, accessibility, and precision of in vivo genome editing in Zebrafish. And second, by combining AB and CB system, we can model both A to G and C to T disease variants in F0 Zebrafish. And third, our platform can enables rapid functional assessment of human genetic variants. So last but not least, I will give my acknowledgement. Thanks all my lab members and my collaborators, Dr. Yamei Liu and Dr. Fang Liang, and the funding. Yeah, thank you.
That was amazing. Let's see when the questions start coming in. I guess I'll ask one in the meantime. How fast is this editing? Like, have you tested when you inject the editor? When do you observe the first editing, from one cell to like, I don't know, like, is it a 512? Is it a 1000 cell stage? Is it a gastrolab? Do you know that?
I don't know the base editor, but some paper reported Cas9 can have the activity, I think, at 4 HPF. Yeah. So I think maybe base editor can also have the activity at that time.
Mm. Let's see. Do you always inject RNA? Have you tried to make protein and see if
Yeah,
it
that's a
works?
good question. Actually, we inject the mRNA, and now the efficiency is very good. And for protein, because it's a little bigger than Cas9, it's not difficult. It's not very easy to purify the proteins. But we tried some proteins from GenScript company, and we found the efficiency can improve further. Yeah, I think the protein is better than mRNA. But
Yeah.
but for us, the mRNA is enough. Yeah.
Is that what is it like almost 10 kilobases long or something, right? Like is humongous the RNA, right?
Yeah. Yeah.
Yeah. Well, I guess if we don't have more questions, I will hand it over to the next moderator. So thank you, Wei. That was fantastic. Thank you.
Yeah, thank you all the attention.
Thank you, A, for the great talk. And hello, everyone. My name is Ece, and I am a graduate student at the University of Texas at Austin. And today I will be moderating the second half of today's session. And I would like to introduce our second speaker, Stefan Choy, who is a PhD student in the Department of Biological Sciences at Lehigh University in Pennsylvania. Stefan is a member of Johanna Kowalko's lab, where he studies the molecular mechanisms underlying behavior using the Mexican cavefish A. mexicanus. Today, he will present his work entitled Identifying the Genetic Architecture Underlying the Evolution of Stress Response in Blind Mexican Cavefish. Please join me in welcoming Stephen.
Yeah, sorry, just sharing screen. Oh. Hi. All right. Thank you for the introduction, and welcome to my talk on. Ooh, sorry, just adjusting some of the settings. Okay. Yes, welcome to my talk on identifying the genetic architecture underlying the evolution of stress response in blind Mexican cavefish. And so, I think it's pretty apparent to everyone that there's wide variations between species. Between zebrafish and humans. We're not quite Actinopterygii, but there's a it's important to remember there's actually a lot of variation within species as well. So humans have a lot of variation, for example, in skin tone. But and a good example are dogs, which have a lot of variation in many different morphological features. But it's also important to remember that there's a lot of variation, not just within morphology, but also within behavior, where two animals from the same species can experience the same stimulus, but have two drastically different behavioral responses. And I think this is again like a very simple concept to understand, but but the underlying molecular mechanisms are really not well understood that underlie this variation. And so one of the central questions that I'm trying to investigate during my dissertation work are what are the genetic underpinnings of complex behavioral traits? And so in order to study this, I use Astyanax mexicanus, which is a single species of teleost fish that exists and multiple different morphotypes. And so we have the surface fish here from the Rio Choy, which you can see on the left, which is very common fish adaptations like an eye, pigmentation, etc. We have multiple cave populations here in the lab from the Pachon, the Molino, and the Tinaja caves. And these fish are well adapted towards their respective environments, whereas surface fish live in different rivers and streams in North Mexico and South Texas, and cave fish live in at least 35 different caves in the North Mexico region. And so these fish are I believe, like a very strong model for investigating the genetic basis of traits for a few different reasons. So one is that these fish are actually still interfertile. These are different populations of the same species. And so we can hybridize them down to the F1 and F2 generations in order to identify the genetic basis of traits. And by both phenotyping these F2 individuals and genotyping these F2 individuals, we were able to perform genetic mapping and identify regions of the genome that underlie the observed traits. And lastly, in Astyanax mexicanus, we also have certain gene editing tools, including CRISPR-Cas9, which allows us to functionally validate any candidate genes that are located within these candidate regions. And so together, this allows Astyanax to study the genetic basis of traits in a comparative manner that's not commonly available to a lot of common model species. And so cavefish have adapted multiple different traits in order to better survive in their environments. And across multiple cave populations, we see many similar traits, excuse me. And so a common trait is a loss of melanin, expansions of the lateral line, loss of eyes, heightened sense of smells, and a larger jaw, just to name a few morphological traits. However, we also see multiple behavioral adaptations, including decreased aggression, decreased stress response, as well as decreased sleep duration. And an open question in evolutionary biology is when you're adapting multiple traits to a new environment, are these traits evolving through similar or shared genetic mechanisms? And so similar or distinct genetic mechanisms, or rather, is it a handful of genes that are contributing towards multiple traits each, or is it multiple genes that each control or each individually dictate one of these traits individually? And so ASCII-NX is a great model to study this because we've already, for the aforementioned experimental pipeline of hybridization into genetic mapping into CRISPR-Cas9 mutagenesis. And so an example of this was the identification of the genetic architecture underlying albinism. And so in several cave populations, albinism, or the complete loss of melanin, has repeatedly evolved. And using genetic mapping, prior work has identified that there was one locus directly over top of the gene OCA2 that had a strong indication that this was causative of the albinistic trait. Using CRISPR-Cas9, we have modified genetically mutated surface fish to have a null mutation at the OCA2 gene, generating surface fish that have only a cave-like mutation at this single locus. And when analyzing other traits, we have identified that previous work has identified that these albino surface fish actually exhibit reduced sleep duration, which is a cave-adapted trait, as well as I've previously demonstrated they have a reduced strike angle, or sorry, an increased strike angle, which is a common cave-adapted trait for larval feeding behavior. And so together, this is a good example that, at least in this case, one gene is influencing multiple cave-adapted traits. But we were interested to see if this is true for other traits as well, and not in other genes as well. And so, my current work is studying is bottom-dwelling behavior, which is the stress response that occurs during a novel tank assay, where when you submit a fish to a new environment, they perform a host of stress behaviors. With the at least most easily quantifiable one is bottom dwelling, where they will inhabit the bottom half of the tank. So I, in previous work, have demonstrated that while surface fish exhibit a high amount of bottom dwelling, so we can see an activity plot on the left, as well as a percentage graphed on the right-hand side, we also see that Pachón, Molino, and Tinaja caves exhibit no direct reduced amounts of bottom dwelling behavior, indicating that this trait has repeatedly evolved across cave populations. And so using the same hybridization into genetic mapping into CRISPR-Cas9 mutagenesis, we will be able to identify the genetic changes that underlie this repeatedly evolved stress response trait. And so this will allow us to ask two important questions. And so the first one is, does stress response share a genetic basis with other cave-evolved traits, like has been previously shown with albinism? And secondly, what regions of the genome contribute towards cave populations' reduced stress response? So in order to study this, our lab, in collaboration with Suzanne McGaw at the University of Minnesota, set up a hybrid cross with surface fish crossed to 3 different cave populations, the Pachón, Molino, and Tinaja again, where we cross them down to the 2nd generation. And in adult hybrids, we submitted them to a 45-day long pipeline where we tested for a host of traits. First off, novel tank, performing the novel tank assay, where surface fish, again, exhibit more bottom dwelling. We measured total sleep, where, again, surface fish exhibit more sleep than cave fish. The total change in sleep after starvation, where surface fish have been shown to sleep less after starvation, whereas cave fish sleep more after starvation. Anesthesia resistance, where cave fish have an increased resistance towards MS222. Eye size and pupil size, where, of course, cave fish have no eyes by the time they are adults. And lastly, the total number of neuromasses located over the SO3 bone region on the face, where cave fish have an expansion of the lateral line. But again, for the purpose of this talk, I'll be mostly focusing on the novel tank assay results. And so first off, here we see the surface fish, which have a lot of bottom dwelling, Pachón, which have a significant reduction in bottom dwelling behavior, F1 surface Pachón hybrids, which have a surface-like amount of bottom dwelling, indicating that the surface allele is dominant in this cross, and F2s, which have a large spread of the trait ranging from surface-like to cave-like. Secondly, for the Molino cross, we see that surface fish, again, and Molino fish have a significant difference in bottom dwelling behavior, but importantly, the F1s for Molino have a intermediate phenotype ranging from an intermediate phenotype that is between surface and merino fish. And the F2s have a spread in the trait, again, ranging from surface-like to cave-like. And lastly, we have the Tanaha population, where surface fish, again, exhibit a high amount of bottom dwelling, the Tanaha have a reduced amount of bottom dwelling, and the F1s actually have a cave-like amount of bottom dwelling, indicating that in this cross, the Tanaha population the Tanaha allele is actually dominant, and the F2s have, again, a spread in the trait. So this is important because it tells us two different things. And so one is that reduced stress response is a multigenic trait. So the spread in the F2s is not following strict Mendelian ratios. It has a range from surface-like to cave-like and everything in between, indicating that in all three cave populations, it's not just one gene or two genes that has been mutated to allow for this reduced bottom dwelling trait. There's multiple genes that all contribute towards the overarching phenotype. And additionally, the modes of inheritance for the F1s are different in all three cave populations, indicating that there are probably different loci that are causing this trait, and that these different loci are changing the dominance patterns across populations. And so next, we took this bottom dwelling trait and we actually performed a Spearman rank correlation test with all of the other traits we analyzed, and statistically significant traits where correlations are highlighted with an asterisk. And so there's a lot going on here, and I think this is interesting but not super useful. And so I've actually compiled this into a correlation summary, where statistically significant correlations are highlighted with either a plus or a minus, depending on the directionality, and the populations are color-coded. And so, for example, just looking at the bottom dwelling correlations, we see that multiple traits statistically significantly correlate across multiple populations with bottom dwelling, indicating that likely bottom dwelling shares a genetic basis with multiple traits. So because these correlations would not likely exist if there was no shared genetic basis. Further, we then compared another trait, albinism, which is a binary trait, which we previously demonstrated, with bottom dwelling behavior and these other traits. And so here we only have Pachon and Merino fish because Tanaha are not albino, and so therefore the resultant F2s are not albino. And we see that when we separate the pigmented versus albino F2 fish, we see that the albinos have a reduced amount of bottom dwelling in both Pachon and Merino populations. Further, the F2s have a reduced eye size. The albino fish have on average, a smaller eye and pupil relative to their pigmented counterparts, indicating that these traits are also correlating or co-varying with albinism. And so this is, again, like a very useful way of investigating whether or not these traits share genetic basis, because we, again, have a stable mutant line for OCA2. And so here, when we reanalyzed these traits in these albino and wild-type surface fish, we see that bottom dwelling behavior is statistically significantly reduced in these albino surface fish, whereas there's no change in eye size or pupil size. And so this gives a strong indication that OCA2 is actually, in addition to pleiotropic impacts on albinism and reduced sleep and larval feeding behavior, it is also impacting stress response, or the bottom dwelling behavior. But it is not directly impacting eye size and pupil size, indicating that there's likely a nearby IQTL or eye locus that is impacting eye size and pupil size in our hybrids, but is not directly OCA2. So next, we then analyzed, so after phenotyping all 1,473 F2s, we also genotyped these 1,400 hybrids in order to generate QTL data, or QTL mapping. And so QTL mapping is a specific type of genetic mapping where we effectively do an association between the phenotype and the genotype at different markers along the genome that we've tracked. And statistically significant, and the higher likelihood that a marker or a loci is contributing towards the trait is represented on the y-axis as the log score, or the log of difference, and the chromosomes are mapped on the x-axis, giving us a map of which regions of the genome are likely contributing towards this trait. And so here you can see between, unfortunately, the Pachonen, we did not have a high enough N for the Merino population in order to perform QTL mapping, as this requires usually around at least 300 individuals, sometimes more. However, we do have it for the Pachonen Tanaha, And we can see that for the bottom-dwelling trait, Pashon and Tanaja have both shared and distinct QTL, which matches the modes of inheritance we saw with the F1s and F2s, where we see that Pashon has QTLs on chromosomes 1, 10, and 20, and Tanaja has significant QTLs on 4, 11, and 20. Importantly, when we looked at, we then QTL mapped eye size, which remember we saw a positive correlation between eye size and bottom dwelling. We see that there is a shared chromosome, a shared QTL for eye size on chromosome 20. Importantly, this occurs in both populations. And when we zoom in just on chromosome 20, we see that this shared QTL is in the same region on both Pashon and Tanaja. So a shared QTL is the same region of the genome in both Pashon and Tanaja is contributing towards both bottom dwelling and eye size, which is a strong indication that this chromosome, this region of this chromosome, is causing that positive correlation that occurred in all three cave populations. Unfortunately, again, we cannot corroborate this in merino fish. We have underneath these QTL, we have 90 genes in Pashon and 23 genes under the Tanaja QTL. And so this is very preliminary, as I've only analyzed the 23 genes, as I've only done a cursory search of the 23 genes underneath the Tanaja QTL. But one of these genes, PTPN11A, in humans causes something called Noonan syndrome, which is characterized by craniofacial defects and eye defects, as well as different behavioral phenotypes, including sometimes in some individuals an anxiety phenotype. And so this is a very strong candidate as we're seeing both eye size and a stress response phenotype within this region of the genome. And so I'm very interested in using CRISPR-Cas9 to target this gene and seeing if there's any phenotype that occurs. And so for future directions, the next thing I need to do is, of course, continue validating the candidate genes that are underlie these QTL, as I've only looked at a very specific region on chromosome 20, but of course there are other regions in other chromosomes on other populations. Additionally, I'm currently performing RNA sequencing on A. mexicanus brains. And so our general experimental design is we have 3 populations that we're analyzing: Zebrafish, Pashon, and Tanaja. We're performing it on both stressed and unstressed fish. And we're dissecting out the brain and separating it into 3 different regions. So the telencephalon, the hypothalamus, and then the remaining brain tissue, which is mostly the hindbrain and the optic tectum. And then ideally we'll generate a list of differentially expressed genes that we'll be able to cross-reference with our candidate genes underneath QTLs from our QTL mapping data to generate a really strong candidate list that are likely causing differences in our stress response trait. And lastly, I'm investigating brain regions that correlate with behavior. And so in addition to our bottom dwelling behavior data of our F2s, which again, we have a range from surface-like to cave-like and everything in between. For a subset of individuals, we've also performed brain dissections, and we've taken out, and so we're able to take out the brain, stain it for a universal nuclear marker, and then we're able to image it on a light sheet microscope, and then we're able to quantify the volumetric data from these different brains. And ideally, we'll be able to take it back and perform, and we're able to correlate region size, brain region size with bottom dwelling behavior in order to identify regions of the brain that likely correlate or contribute towards differences in bottom dwelling behavior. And so in conclusion, going back to the 2 major questions, so does stress response share a genetic basis with other cave evolved traits? And so I've demonstrated that yes, it does. So we have multiple correlations for bottom dwelling behavior for multiple different traits that we measured. And when using a very specific when using specific stable lines for mutations that for cave-like mutations, we've demonstrated that bottom dwelling behavior is specifically impacted through the albinism gene OCA2. And to answer the second question, what regions of the genome contribute towards cave populations' reduced stress response? Using QTL mapping, we've identified multiple regions of the genome that statistically significantly correlate with bottom dwelling behavior, including a shared QTL in 2 different populations, indicating potential repeated evolution, or a QTL that was present in the original surface-fish population that has been inherited by both Pashon and Tanaja populations, but that again warrants further further investigation. Again, on chromosome 20, and so I'd like to give thanks to Joanna Kowalko, my PI, of course, as well as several other lab members that have been very useful and very helpful on this project. Aubrey Manning and Brooke Coonrod, as well as the McGaw Lab, who we could not have performed this large-scale project without, and they've been very helpful for the QTL mapping data as well as general sequencing. And I'd like to give thanks to the Department of Biological Sciences, including the chair of the department, Kathy Iovine, and the staff members, Aaron Curtolo, Lee Graham, Katie Stevens, and Lisa Sidlow. Thank you. Any more questions? Any questions?
Thank you, Stefan, for this great talk. So we will wait some Q&As and I can update you. But I actually have a question. Maybe it's like a little bit unrelated, but like for example, like pigmentation can be
Yes.
also caused by high level of cortisol. So there's actually a link between a stress level versus like melanin production, at least in human. And like maybe these kind of things are also correlated in like a different way of like increase in melanocyte productions. So like
Yes.
so do you think that these all phenotypes that you see is like correlated in one basket, or do you think that there are different mechanism, it just in the correlation level but not causation level in that case?
So there is definitely within Astyanax the ability to modulate the number of it's either the number or the size of the melanocytes. So for example, when you submit them to MS222 or Tricane, the melanocytes will bloom. And so how you quantify melanocytes is or melanophores, excuse me, in fish is different because you need to be able to very carefully quantifying them is difficult because they change size and can kind of blend together when you present them with in the presence of anesthetics. However, OCA2 prevents the formation of melanophores and melanocytes at it stops it at the first step. So OCA2 is a I believe a sodium channel transmembrane protein present on melanophores, and mutations in that either cause, in this case, a frame shift, or in Paschon and Molino, there's exon deletions, prevents the formation of melanophores in the beginning and in the first place. And so while cortisol may be impacting pigmentation, or there may be a like a non-traditional correlate like a non-causational correlation, I would suspect that it's likely due to other factors than directly some pigmentation melanophore correlation, because these fish just simply don't have these melanophores.
Yeah.
Does that answer your question?
Yeah, I think so. Yeah, definitely. It's an interesting observation, definitely. It's a great talk.
Thank you.
So let's see. We will wait for maybe 1 more minute for the questions, and after that, I will hand over for closing remarks.
Okay.
I actually have a question while we wait for people to type in the chat. So do you think that the eye loss and the behavioral traits that you're seeing evolved because they're genetically linked, or during selection independently favored, where each of these like independently favored and now they happen to.
There's a ongoing question in evolutionary biology about the importance of pleiotropy. So traditionally, like going back to Ronald Fisher in the 1920s and 1930s, postulated that pleiotropy was not favored by evolution because one mutation now is like deleteriously impacting two traits. But what previous work, and especially in sticklebacks, as well as in previous work in Astyanax, has shown that when you're adapting to a new environment, you need multiple traits to adapt. You're not just changing one trait, you're changing multiple. And so if you serendipitously pick up a mutation in a pleiotropic loci that impacts two traits in the direction that you want, so again, like for example, OCA2 is a great example because like albinism is a repeatedly evolved cave adaptation. This is seen across taxa. Eye size is impacted, or sleep is another trait that's impacted, right? And so if you need to evolve both reduced sleep and albinism, then when you pick up mutations in OCA2, it's selected for at a higher rate than picking up individual mutations in just one of those traits, right? Because now you're essentially two birds with one stone. And so there's, I think, a bigger impact of pleiotropy on multi-trait evolution than people originally gave credit for. And I would suspect that as we continue to analyze both Astyanax, but other examples of repeated evolution or multi-trait evolution, I'm sure we'll see more pliotropoglossi coming into play. Does that quite answer your question?
Yeah, no, that's great. It's very interesting stuff. So thank you.
Oh, thank you.
Okay, I think we don't have any questions in the chat, so we
Okay.
can go for the closing remarks.
Awesome. Well, my name's Shannon. I'm one of the York board members. I just want to thank you all for attending. I want to thank Wei and Stefan for giving some great talks today. And of course to the moderators for helping or assisting during this session. So a recording of today's webinar will be available on the IZFS website, and it'll be on our York YouTube page within the next few days or so. We'd also like to invite you to join us on our next FPK seminar, which will be on September 24th. And so during that one, we'll hear from Shuyu Zhu, who's a postdoc at Washington University in St. Louis, and Samudra Gupta, who is a PhD student at the University of Calcutta. And with that, thank you all for attending, and we look forward to seeing you next time.